This is lecture 14 for regulation of blood pressure. Looking at the regulation of blood pressure, we have already covered blood flow through the heart and some of the basic anatomy of the blood vessels. What we're seeing here is simply a functional model of the cardiovascular system that allows us to clearly see the circulation of blood through the body. The vascular system moves blood throughout the body through two distinct circuits: the pulmonary circuit, which is the lungs and respiratory center, and the systemic circuit, which is through the body and the brain. We can see starting at the left side of the heart, where it goes through the left ventricle and out through the aorta, it will then pump through the aorta and through the arteries and then eventually enter into the arterioles. The arterioles, shown with little adjustable screws there, are shown in that way so that you can see that they alter their diameter and are the site of variable resistance, and this allows you to control blood flow to different capillary beds. The capillary beds are going to be where exchange happens between the blood cells and the actual cells of the tissue itself. From there, it will move into the venules as it travels back through to the right side of the heart. A key aspect of both arteries and veins is that they are reservoirs. Arteries themselves are elastic systems and pressure reservoirs. This helps it to maintain a high pressure throughout the system to allow blood to flow from the direction of arteries towards veins. Veins themselves, not being a pressure reservoir, are a volume reservoir. This means more of the overall volume of blood is going to exist in the actual veins as opposed to the arteries. This higher volume of blood allows it to gradually travel its way back to the heart, affecting rate of venous return. Just as a reminder from a previous lecture, we have a very unique structure called the vasovasorum. The vasovasorum is going to be on the tunica adventitia, which is the outer connective tissue layer on all blood vessels, but the vasovasorum is going to be found only on large arteries or veins, specifically the aorta and the vena cava. This is going to be a series of blood vessels that actually supplies nutrients to the blood vessel itself, because these vessels are so thick that they cannot obtain nutrients directly through diffusion in the lumen. Here we can actually see the layout and the layering of blood vessels as discussed in previous lecture. We can see the innermost layer of the tunica intima, which is going to be the innermost layer, the tunica media, which is the middle layer, and the tunica adventitia, which is the outer or connective tissue layer. I apologize, there is an error here in the labeling of tunica intima and tunica media. You can flip those layerings on this diagram to make it correct. Moving into portal circulation systems on your lecture notes, we can see here is a basic diagram of the portal circulatory route. In many cases throughout the body, it goes arteries, capillaries, and veins. However, with the portal circulatory route, it goes artery to capillary bed, portal vessel, capillary bed, and vein. Now, the purpose behind a portal vessel is to direct flow. There are three special circulatory patterns in the body that have two capillary beds in the series. These are going to be the hypophyseal hypothalamic portal system that we discussed in endocrine, the renal portal system, which would be in the kidneys, and the hepatic portal system that is in the liver. Here we can see a simple functional layout of these different capillary beds. The aorta would head down through the hepatic artery and into the liver. This would be a normal blood flow of going artery, capillary to vein. As that aorta connects down to the splenic artery and to the mesenteric arteries, we can see that it goes through a capillary bed here and then into the hepatic portal vein. The hepatic portal vein itself is actually going to be a connection from the different digestive blood vessels into the liver. The liver is the only abdominovisceral organ that drains into the inferior vena cava. All of the other digestive organs drain first into the hepatic portal system to regulate the process and nutrients and toxins from the GI tract. Here we can see a diagram of the hepatic portal vein and some of the connections that lead directly into it. We can see the mesenteric, both the inferior and superior mesenteric veins, as well as the splenic vein, as they all connect to the hepatic portal vein. The three major veins that drain to the hepatic portal vein are the superior mesenteric vein, the splenic vein, and the inferior mesenteric vein. These are going to drain directly to the hepatic portal vein, which thereby drains into the liver to allow for filtration. And from the liver, it will go through the hepatic veins into the inferior vena cava. The portal systems are vital in making sure that we can isolate blood flow to either, in the case of the hypophyseal portal system, prevent dilution, or in the case of the hepatic portal system, to ensure filtration and to protect the body from something that we might have ingested. This allows us to filter out different sorts of poisons or toxins that we might consume in the diet, like different drugs or alcohol. And it also allows us to properly process proteins, fats, and carbohydrates and synthesize them into simpler nutrients that our body can more readily use. From the hepatic portal system, I want to discuss blood pressure. Now, blood pressure is initiated from the ventricles. The first form of this starts when the ventricles contract, specifically when the left ventricle contracts. The left ventricle contracts with pressure increasing above that of the arteries. From here, the semilunar valve will open and blood becomes ejected, flowing out into the arteries. The aorta and the arteries will expand and stretch while blood and pressure force its way out, and this stretching causes storage of elastic energy. This is like pulling the bowstring back on a bow. You're storing elastic energy in this elastic cord, and then when you release it, it fires, releasing that elastic energy. We can see the release of this elastic energy as during the isovolumic ventricular relaxation phase, the semilunar valves shut. This prevents backflow into the ventricles. A natural flow of blood back against the walls of the arteries is going to then force an elastic recoil. This elastic recoil is going to send blood forward into the circulatory system, causing that pressure gradient. Blood pressure is going to look at systolic and diastolic pressures. As always, remember that systolic pressure is going to be the pressure during contraction. Diastolic is going to be the pressure during relaxation. Subtracting the systolic minus the diastolic pressure gives us what we call pulse pressure. Blood pressure is measured in a lot of cases through the pulsatile in the arteries. This means that blood flow and pressure increase and decrease with the cardiac cycle, and this gives rise to arterial pulse pressure. The pulse pressure, as stated, is going to be the systolic minus the diastolic pressure. This pulse is most easily felt or measured in large arteries, specifically places like the carotid artery, which would be in your neck, the femoral artery in your thigh, and then in the radial arteries as well, that would be on the thumb side of your wrist. This pressure is going to disappear as the blood enters the capillary beds. Now, let's look at what happens to blood pressure through the circulatory system. Blood pressure through the entire circulatory system is going to be very high in the arteries and arterioles and then get gradually lower as we move throughout. Arteries have a very clear pulse pressure. This is a transition from diastolic to systolic as the heart contracts, and we can see that here. The systolic pressure is going to be the high-end pressure, the highest pressure that the artery is at. The diastolic pressure is going to be that pressure that it actually is going to drop at, where it relaxes. When we look at measuring blood pressure is often referred to as one number over the other, with an average healthy blood pressure of being 120 over 80. This is actually going to be the top pressure, systolic, and the bottom pressure, diastolic. These two numbers together would equal your pulse pressure. We can see on the line as it extends down, we get mean arterial pressure. Now, mean arterial pressure is going to be diastolic pressure plus 1/3 of the pulse pressure. This is simply looking at the average arterial pressure of the system. You will not have to calculate this on the exam. You will not have to calculate pulse pressure either. As this extends down, we can see that pressure drops markedly when it gets to the capillaries. In the capillaries, we have no pulse, and the pressure has decreased steadily. This is due to increased cross-sectional area in the capillaries, a lot of space for a lot of blood to move through. We also have a decrease in pressure in this area in order to help facilitate a better rate of diffusion. From there, we then enter into the venules and the veins. We cannot feel a pulse in the venules and veins as there is no arterial stretch because they're not arteries. And then the pressure will continue to decrease as it gets towards the right atrium. It is almost near zero pressure when it gets to the very end of the vena cava, but pressure never fully drops to zero. And that is an important thing to note. Note from the previous slide that blood pressure is above zero everywhere in the vascular system. What keeps blood flowing in the low pressure venous system? There are three factors. As can be seen in the image here, one of the major factors is that in lower extremities, larger veins have one-way valves to keep blood flowing upwards. Veins can also contract around blood, increasing the pressure slightly and forcing blood to flow. Here we can see an example of how the skeletal muscle pump actually works in squeezing the blood vessel and forcing blood up through the valves, ensuring a one-way system transferring blood back to the heart. The final reason is because the blood is still flowing from areas of high pressure to low pressure, so we always have that gradient apparent. So, mean arterial blood pressure is affected by a number of variables. The blood pressure overall is maintained above zero throughout the circulatory system, except maybe in the vena cava and the atria and ventricles where it drops down a little bit. This is primarily because the arterial system is elastic and maintains pressure around the blood. It is difficult to measure blood pressure in the ventricles without invasive procedures, so clinicians measure pressure in the arteries instead. This metric is called the mean arterial pressure, as has been discussed. Note that the mean arterial pressure is closer to the end of diastolic pressure than to peak systolic pressure. This is because diastole lasts about twice as long as systole. Another way to think about mean arterial pressure is that it is a function of cardiac output and arterial resistance, and we can see that here. We have cardiac output, which is determined by both heart rate and stroke volume. We have the overall blood volume that is going to affect the volume of blood will actually affect blood pressure because of the natural hydrostatic pressure of fluid forcing its way up against the walls of its container. Blood volume is determined by both fluid intake and fluid loss. Fluid loss can be passive, simply through the skin, or it can be regulated at the kidneys. Other variables that affect mean arterial blood pressure are going to be the resistance of the system to blood flow, which is determined by the diameter of the arterioles, and the relative distribution of blood between arterial and venous blood vessels. This is determined by the diameter of the veins. Now that we've talked a little bit about mean arterial pressure, I'd like to talk about the regulation of blood pressure. What are the factors and structures that actually regulate this? This diagram might seem complicated at first, but what we can clearly see is that there's going to be, in the yellow circle, a stimulus that would be a change in blood pressure. This stimulus will be felt by specific sensory receptors, the carotid and the aortic baroreceptors. If you remember from our lectures in the nervous system, baroreceptors are pressure detection receptors. So they are going to detect the stretch in the arterial wall and the increase in pressure. This information is going to be sent up towards the brain at the medullary cardiovascular control centers. From here, it can affect different changes in the autonomic nervous system in both the sympathetic and parasympathetic neurons. This can cause changes in either heart rate, in the case of just the parasympathetic, or it can affect heart rate, ventricular force contraction, and arterial and venous contraction in the case of the sympathetic nervous system. Let's take a look at an example of how this works. Here we can actually see a nice flow chart with a stimulus and the overall response that it would have. In this case, the stimulus that we have is an increase in blood pressure. That increase in blood pressure is going to then cause an increase in the firing of baroreceptors in the carotid arteries and aorta. This means that they're detecting an increase in pressure. It simply increases firing, so it's going to actually just start to turn on those sensory neurons, thereby communicating this information to the cardiovascular control center in the medulla oblongata. The medulla oblongata will have two overall effects. It's going to have an effect on the sympathetic end and on the parasympathetic end. I'll start with the parasympathetic end. the parasympathetic output is going to increase. What this means is the parasympathetic output is going to affect the SA node to decrease heart rate. That's what the parasympathetic innervation does, as we discussed in previous lectures. This decrease in heart rate will cause a decrease in cardiac output. If cardiac output goes down, blood pressure goes down. We saw this before with the overall formula of mean arterial pressure. Now let's go over to sympathetic output. Sympathetic output will decrease. This decrease in sympathetic output causes arterial smooth muscle to dilate, causes the force of heart rate contraction to go down, and allows the heart rate to decrease. The decreasing, or the vasodilation rather, is going to cause a decrease in peripheral resistance. The decrease in force of contraction causes a decrease in cardiac output. These decreases, again, combined with the decrease in heart rate, is going to decrease blood pressure. As blood pressure goes down, we then have a negative feedback loop that signals to the baroreceptors, blood pressure is fine, no need to continue. So this is going to be an important reflex that regulates blood pressure for the short term, and eventually it accommodates to chronic changes in pressure. But again, this is a short-term response. Long-term regulation is a function of the kidneys, and we'll look at that a little bit later.